Hi guys, welcome to our discussion on Chapter 13, Section 3, Meiosis. This is going to walk through the actual process of meiosis and compare it a little bit to mitosis to see the similarities and differences there. So to begin, let's review like mitosis. Meiosis is preceded by the replication of chromosomes. Meiosis takes place in two sets of cell divisions, meiosis 1 and meiosis 2. The two cell divisions result in 4 daughter cells rather than 2 daughter cells in mitosis. Each daughter cell has only half as many chromosomes as the parent cell. Right? In the first cell division, meiosis 1, your homologous chromosomes separate. Meiosis 1 results in 2 haploid daughter cells with replicated chromosomes. It is called the reductional division. In the second cell division, meiosis 2, the sister chromatids separate. Meiosis 2 results in 4 haploid daughter cells with unreplicated chromosomes. It is called the equational division. Okay, let's take a peek at the overview of meiosis and how it compares to mitosis. Through interphase, you begin with a homologous pair of chromosomes in your diploid parent cell. The chromosomes replicate, giving you a homologous pair of chromosomes that are paired with their sister chromatids. Okay? So your end of interphase, your diploid cell with its replicated chromosomes. Into meiosis 1, your homologous pairs separate into new cells. Now you have haploid cells with the replicated chromosomes. Okay. Meiosis 2 separates those sister chromatids into each their own cell, and this leaves you with 4 cells that have half the genetic material of the parent cell, each being genetically different, having some variation based on what genes it inherited. Okay, if you want to take a closer look at this figure, I highly recommend it. It's figure 13.7 on 253. Make sure you understand this overview so that when you walk through each individual stage of meiosis, it will make more sense. You see where you're heading here. Okay. Meiosis 1 is preceded by interphase, in which chromosomes are replicated to form sister chromatids. What we were just looking at up here. The sister chromatids are genetically identical and joined at the centromere. The single centrosome replicates, forming 2 centrosomes. Okay. This figure, 13.8, is found on 253 or 254 and 255 in your textbook. I highly recommend that you sit down and really study through this figure and walk through what's occurring at each phase. I believe that would be extremely beneficial. Okay. Alright. Division in meiosis 1 occurs in 4 phases: prophase 1, metaphase 1, anaphase 1, telophase 1, and cytokinesis. Alright. Prophase 1, metaphase 1, anaphase 1, telophase, cytokinesis. Just as what we looked at in the previous figure, the goal of meiosis 1 is to separate your homologous pair. So, at the beginning of prophase 1, you have your replicated chromosomes that are still matched up with their homologous pair. Throughout the meiosis 1 process, your homologs are split into new cells, divided in half. Okay? But they are still replicated and still paired with their sister chromatid. It's important to note that replication only occurs at the beginning of meiosis 1. It does not repeat as it goes into meiosis 2. Okay? Prophase 1 typically occupies more than 90% of the time required for meiosis. Your chromosomes begin to condense. In synapsis, your homologous chromosomes loosely pair up, align gene by gene. Okay. In crossing over, non-sister chromatids exchange DNA segments. That's why when you look back at this figure, you see there are little red sections or blue sections on these new chromosomes. That's representing crossing over. Okay, exchanging of genes. There it is, crossing over. It exchanges that DNA segment. Each pair of chromosomes forms a tetrad, a group of 4 chromatids. Each tetrad usually has one or more chiasmata, the X-shaped region where crossing over has occurred. Alright, in metaphase 1, your tetrads line up in the metaphase plate with one chromosome facing each pole. Microtubules from one pole are attached to the kinetochore of one chromosome of each tetrad. Your microtubules from the other pole are attached to the kinetochore of the other chromosome. Okay, getting everything lined up, ready to separate. Alright, here showing that stage. Anaphase 1, pairs of homologous chromosomes separate. One chromosome moves toward each pole, guided by the spindle apparatus. Spindle chromatids remain attached to the centromere and move as one unit toward the pole. Alright, telophase and cytokinesis. In the beginning of telophase 1, each half of the cell has a haploid set of chromosomes. Each chromosome still consists of those 2 sister chromatids, and cytokinesis usually occurs simultaneously, forming 2 haploid daughter cells. They still have the sister chromatid is still replicated here. Make sure you note that. In animal cells, like with cytokinesis in mitosis, the cleavage furrow forms. In plant cell, the cell plate. So the cytokinesis remains the same process. There is no new replication between the end of meiosis 1 and beginning of meiosis 2. Because your chromosomes are already replicated, there's no need to replicate them again. Okay? Here showing anaphase telekinesis one more time. Telophase cytokinesis. Alright, division in meiosis 2 also occurs in 4 phases. Prophase 2, metaphase 2, anaphase 2, and telophase 2 and cytokinesis. Meiosis 2 is very similar to mitosis. Okay, here's showing that process for you. I will let you dig in on your own to make sure you understand this process, okay? Like always, if you have questions, let me know. Alright, to overview prophase 2 very quickly, I won't linger much because this is detailed out pretty nice and neat in your notes, in your book. We will discuss this Friday when we have some time in class, okay? Prophase 2, a spindle apparatus forms. In late prophase 2, chromosomes, each still composed of 2 chromatids, move toward the metaphase plate. In metaphase 2, the sister chromatids are arranged at the metaphase plate. Because of crossing over in meiosis 1, the 2 sister chromatids of each chromosome are no longer genetically identical. The kinetochores of sister chromatids attach to the microtubules, extending from the opposite poles. Alright, here you are. Everybody's lined up, ready to separate back to into their new home. In anaphase 2, the sister chromatids separate. Sister chromatids of each chromosome now move as newly individual chromosomes toward opposite poles. Telophase 2, the chromosomes arrive at the opposite poles. Nuclei form and the chromosomes begin decondensing. Cytokinesis separates the cytoplasm. At the end of meiosis, there are 4 daughter cells, each with a haploid set of unreplicated chromosomes. Each daughter cell is genetically distinct from the others and from the parent cell. Alright, here's showing anaphase 2, telophase 2, and cytokinesis. Alright, a comparison of mitosis and meiosis. Remember that mitosis conserves the number of chromosome sets, producing cells that are genetically identical. Meiosis, however, reduces chromosome number. Instead of having a diploid cell, you end up with a haploid cell, and they are genetically different. Sorry, guys. They're genetically different from each other and from the parent cell. Alright? The mechanism for separating sister chromatids is virtually identical in meiosis 2 and meiosis, and mitosis. So, meiosis 2 and mitosis, the process itself is very, very, very similar. Okay? Here, showing you a side-by-side comparison of meiosis versus mitosis, showing your prophase area, metaphase, anaphase, and telophase. Go to meiosis 1, and then the end of meiosis 2, and you end up with 4 haploid daughter cells. Okay? I would definitely look back over this too. This would be something I'd be able to conversate on. If you had a free response question on this, this would be one that you need to be able to explain where they're similar and where they differ. In both of these examples, the parent cell is 2n equals 6, so it has 6 chromosomes. Meiosis, your chromosomes are in homologous chromosome pairs. Going through metaphase, your homologous pairs line up, and your homologous pairs are what are split into what's going to be your daughter cells. So then, when you go into meiosis 2, those sister chromatid pairs are then split. So it just takes that extra set of division to get the haploid state. Okay? Here's showing that again, just looking up close. I do recommend you take a peek at this. Okay? There are 3 events that are unique to meiosis, and all 3 occur in meiosis 1. Your synapsis and crossing over in prophase 1, your homologous chromosomes physically connect and exchange genetic information. Crossing over is a big part of meiosis. That gives you genetic variety. Alright, at the metaphase plate, there are paired homolog chromosomes, your tetrads, instead of individual replicated chromosomes. And at anaphase 1, it is homologous chromosomes instead of sister chromatids that separate. Okay? So your key things to note different-wise. Sister chromatid cohesion allows sister chromatids of a single chromosome to stay together through meiosis 1. Protein complexes called cohesions are responsible for this cohesion. In mitosis, cohesions are cleaved at the end of metaphase. However, in meiosis, cohesions are cleaved along the chromosome arms in anaphase 1, so separation of your homologs, and at the centromeres in anaphase 2, separation of the sister chromatids. Okay, this expanded in your book is quite interesting. It talks about how the cell knows which cohesion to cleave, and it comes down to, let me double check myself. It comes down to a specific protein that they gave a name that is Japanese for guardian spirit. Alright, this guardian spirit protects your cohesions from cleavage at the centromere during meiosis 1. It's this specific protein that is in place so that it is not fully cleaved at meiosis 1, but is able to carry on to meiosis 2. Okay, we're going to stop here for 13.3. That is a wrap. Make sure to answer your checkpoint questions, and we'll continue with 13.4. Thanks.